Why do some reactions happen instantly while others take forever? What actually controls the speed of a chemical reaction? The answer is enzymes. And in this video, we're going to cover and break down enzymes for AP biology, what they are, how they work, and why they're so important. So what are enzymes? Enzymes are proteins that act as biological catalysts. They will speed up chemical reactions without being consumed. And this is important because it would mean that a single enzyme can be reused again and again to help reactions occur faster. Here are some key terms that you should know. The molecule an enzyme acts on is called the substrate or the reactant. The substrate binds to a specific region on the enzyme known as the active site. The enzyme-substrate complex is the temporary molecule formed when the substrate is binded to the active site. After reaction, the substrate is converted into products. Because enzymes speed up reactions without being used up, they're considered catalysts. There are two main models that explain how substrates bind to enzymes. In the lock and key model shown up here, the substrate fits perfectly into the enzyme's active site. In the induced fit model shown down here on the right, the enzyme slightly changes shape as the substrate binds, which will improve the reaction. In both cases, an enzyme-substrate complex forms, the reaction occurs, and the enzyme is released unchanged, so it can be reused in a different reaction. Every chemical reaction requires activation energy, which is the energy needed to start the reaction. Without enzymes, this energy barrier is high, so the reactions happen very slowly, as represented by the purple line over here. Enzymes will lower this activation energy, which will make it easier for reactions to occur, and increase the reaction rate, shown as the green line. There are several factors that affect enzymes. Temperature affects how fast molecules move. Too high will denature the enzyme, and too low will slow down reactions because there will be less enzyme-substrate complexes formed. pH will also matter because changes in pH can alter the enzyme's shape and cause denaturation. The substrate concentration affects how often enzymes and substrates collide. The enzyme concentration will also do the same, but if the substrate is steadily available, it will be constant. But on the other hand, if the substrate is limited, it will even out, such as the line in the substrate concentration. Inhibitors are also a very important part that slow down reaction rate. Competitive inhibitors bind to the active site itself and compete with the substrate. Non-competitive inhibitors will bind to a different location on the enzyme, often known as the allosteric site, and change the enzyme shape, which will alter the shape of the active site and make the substrate unable to bind to the enzyme. Both of these will reduce the rate of the reaction. Cofactors and coenzymes. Some enzymes require additional helpers to function properly. Cofactors are often inorganic ions such as magnesium. Coenzymes are organic molecules often derived from vitamins like NAD+. Without these helpers, some enzymes cannot catalyze reactions. As seen here, in the specific example, the cofactor is a coenzyme. The substrate initially cannot bind to the active site, so what happens is the cofactor or the coenzyme will bind to the active site in case of the substrate, and this will cause a conformational change in the shape of the enzyme. This will make it a better fit for the substrate, where the enzyme can convert the substrate into products. Here's an AP style question for you. ALAD is an enzyme that relays on zinc as a cofactor. A zinc ion binds to ALAD active site where it forms favorable interactions with side chains of 3 amino acids. Researchers have found that substituting a lead ion for a zinc ion in the ALAD active site causes inhibition of ALAD. Which of the following statements best explains how the lead ion causes inhibition of ALAD? A. It changes the shape and charge of substrate so that it becomes more compatible with ALAD's active site. B. It changes the amino acid sequence of ALAD protein so that the enzyme catalyzes a different reaction. C. It changes the three-dimensional structure of the active site so that ALAD is no longer compatible with the substrate. Or D. it changes the enzyme substrate complex so that the transition state is more stable and the reaction proceeds at a faster rate. Please pause the video here and come back when you have an answer. So the correct answer is C. The substitution of a lead ion for a zinc ion will most likely change the three-dimensional structure of the ALAD active site. The three-dimensional structure of the active site plays a very important role in the actual compatibility between an enzyme and a substrate. So changing the structure of the site will interfere with the enzyme's function by disrupting the enzyme-substrate interaction and reduce the reaction rate. Here's another question for you. An enzyme is placed in a different container. An enzyme is placed in different containers with differing temperatures. Explain why the reaction rate looks like this. The optimum temperature is 40 degrees Celsius, and to the left there's a steady decrease, and to the right there's a steep decrease. Please pause the video here and come back when you have an answer. So your answer should look something like this. To the left of 40 degrees Celsius. Since the temperature will decrease, less kinetic energy is involved and less enzyme substrate complexes will be formed. This in turn will result in a steady decrease in reaction rate as temperature decreases. To the right of 40 degrees Celsius, the extreme temperature will denature the enzyme, and I'm looking for this word here, and lead to unusable enzymes. This will result in a sharper decrease in reaction rate. Here are some key takeaways from this video. Enzymes speed up reactions. Structure determines function. Environment affects activity. And enzymes are specific to their substrates. Thank you.